So a plasma membrane has many different functions, and they're accomplished with the help of different types of protein, carbohydrate, and lipid molecules that are embedded within or attached to the phospholipid layer. Many of these molecules float around, and they're held in proper orientation by the hydrophobic and hydrophilic forces. But they're not always anchored in place. For these reasons, the plasma membrane is often described as a fluid mosaic. For every 50 to 100 phospholipids that we find in the membrane, there's one protein molecule. Some of the proteins, called transmembrane proteins, penetrate right through the lipid bilayer from one side to the other. Others, that are called surface proteins or peripheral proteins, reside primarily on the inner or outer surface of the membrane. Whether a protein resides on the surface or extends through the bilayer is its tertiary structure. So remember from an earlier lesson that all the amino acids that make up each protein have side chains that differ from one another chemically, and some of these side chains are hydrophobic while others are hydrophilic. And as a protein is assembled into its final shape, these side chains can cause parts of the protein to be attracted to the hydrophilic regions of the molecule. Because a transmembrane protein has both hydrophobic and hydrophilic regions, part of the protein can be positioned in the hydrophobic region in the center of the membrane, while the other parts are in the hydrophilic regions. Surface membrane proteins, on the other hand, have an entirely hydrophilic structure, and so they reside on the membrane surface, not only of the phospholipids. As a consequence of that, they can be positioned on the outer or the inner surface of the membrane. Once membrane proteins are in place, hydrophobic and hydrophilic forces keep them properly oriented. And because all of the components of the plasma membrane are held in the membrane in this manner, they can float around without ever popping out. There are several primary types of membrane proteins, and each of them performs a different function. The first one is receptor proteins, and they're a surface or transmembrane protein that bind to chemicals in the cell's external environment. When they do this, receptor proteins can attach a cellular matrix or convey information from the outside to the inside of the cell, regulating certain processes within the cell. This receptor-mediated regulation is a process called signal transduction. Cells in the heart, for example, have receptor proteins that bind to adrenaline, a chemical that's released into the bloodstream in times of extreme stress or fright. When adrenaline binds to these heart cells, cells increase the heart rate of contraction to pump blood through your body more quickly. You've experienced this reaction if you've ever been startled and felt your heart start down. The second one is recognition proteins, and they're surface or transmembrane proteins that give each cell a fingerprint that makes it possible for the body's immune system, which is fight infections. It allows the body's immune system to distinguish the cells that belong inside your body from those that are invaders and need to be attacked. Carbohydrates also play a role in recognition. Glycoproteins, or sugar proteins, on the outside of the cell membrane serve as part of the membrane's fingerprint. Like receptor proteins, recognition proteins can also help cells bind to or adhere to cells or molecules within the extracellular matrix. The third type is transport proteins, and they are transmembrane proteins that help polar or charged substances pass through the plasma membrane. Transport proteins come in a variety of shapes and sizes, making it possible for a wide variety of molecules to be transported. The fourth is membrane enzymes, and they are surface or proteins that help accelerate chemical reactions on the plasma membrane surface. A variety of membrane-bound enzymatic proteins exist, with some accelerating reactions on the inside of the plasma membrane and others accelerating reactions on the outside of the plasma membrane. You can see these four types of proteins in the image shown. In addition to the various kinds of membrane proteins and the carbohydrate chains of membrane-bound glycoproteins, the lipid cholesterol can also be incorporated in a cell's plasma membrane. Cholesterol helps the membrane maintain its flexibility, preventing the membrane from becoming too fluid or floppy at moderate temperatures and acting as sort of an antifreeze, preventing the membrane from becoming too rigid at freezing temperatures. The membranes of some cells are about 25 cholesterol. Other plasma membranes, such as those of most bacteria and plants, have no cholesterol at all.